Okay, welcome everybody. Today we have a webinar with Stephen Russell, the next big thing in mycology. I'm pretty excited to kick it off. This is Trent Blizzard, and I'm the president of NAMA this year. And I think before we get going with Stephen, I want to do a couple of house cleaning issues. Number one is if you have any questions during the presentation, use the Q&A tool in the Zoom meeting, and you can post your questions in there. Stephen's going to present for about an hour, and then he will go and start working through your questions, and he has at least a half hour to really try to answer as many good questions as you all are able to submit. So that should work well. We are recording this session, and it will be posted up on our website and our YouTube channel probably in the next 2 days. And if you go to the website and look under webinars, you'll see all our previous webinars up there. I'm excited to announce we have 2 upcoming forays. Number one, we have a new foray. It's going to happen in Mexico, and that's going to kick off July 30th. And we are probably going to announce that to the world, we hope, on Monday. So watch your emails. It might be a day sooner, might be a day later. But it's going to be a really cool regional foray base. We'll fly into Mexico City. We'll visit some mycologists or herbariums there, and then head forthwith up into the mountains, or I should say over into the mountains, maybe there, and have a 4 or 5 days of foraying there. It's going to be an exciting event. And we have our annual Appalachian NAMA coming up. That's going to be in North Carolina, outside of Asheville, and that will be at the end of August. We'll be announcing that. We're probably a month or 6 weeks out from opening registration on that, but watch, we will have announcements coming on that. And we do have an upcoming webinar, February 8th. We have Rachel Sweeney, who's going to present to us on chanterelles, which chanterelle is the name of her presentation, and she's going to interpret the latest North American chanterelle research. So that's an exciting presentation. Now about Stephen, I met Stephen officially right here. This is a cabin at the last NAMA event, and Stephen is over there on the couch, the third from the right. Next to him is Mandy, and I see Gabrielle, and is that John maybe back there in the chair? I'm not sure I recognize everyone else from here. However, you can see his equipment there, and he came out to our annual foray and brought his laptop and all this crazy cool testing equipment, and he ran tests on, I think, literally hundreds, maybe 200. I don't know exactly. He'll tell us. Specimens right there in his room, and his computer was churning away, I think, for 48 hours or more. It was pretty exciting to watch the science happening right there in that cabin, which was next door to my cabin. So I kind of got to check in on it pretty regularly. So that's where I met Stephen. But to kind of give you a little bit more official details about him, he's a mycologist working in Indiana, where he's really been specializing in the biodiversity survey aspects and doing a lot of DNA-based work out of there. He founded the Hoosier Mushroom Society in 2009, and he is the president of that organization currently. And he's also well known for writing a really great book on cultivating, The Essential Guide to Cultivating, back in, I guess, 2014. The Essential Guide to Cultivating Mushrooms, back in 2014, which is a pretty well-received book, and if you cultivate, is probably in your library already, I'm guessing. He chaired the NAMA Mycoflora Committee and was also the co-founder of the North America Mycoflora Project. Current project is citizen science and DNA-based exploration. He has a pretty cool BioBlitz going on right now. And we have a few more, or he has a few more scheduled for later in the year that hopefully NAMA is going to get involved in. So you'll be seeing, I think, a lot of him this year as he works to DNA sequence a lot of mushrooms and really enlarge his collection of records with over 2,500 unique species, which is just crazy cool. So I'm excited to turn this over to you, Stephen, and have you take over. I'm going to turn my video off and welcome you and look forward to sitting back and watching your presentation.
Yep, thanks a lot, Trent. We were talking a little bit before this presentation is about to start about the cultivation book. Something that I don't think I've ever shared with anybody actually is that book actually got started writing it while I was waiting at a bus stop at the end of the Telluride Mushroom Festival in I think 2013. And so I actually burned through 2 chapters while I was sitting there waiting on a bus, just, you know, longhand in a notebook. And so the first couple chapters from that were actually written at the end of the Telluride Mushroom Festival the previous year. Let me minimize this window if I can. That was the wrong button. I hope you guys can see my screen currently, the screen share, and I lost all of my Zoom functionality. If I need to mute, okay, there we go. So Trent gave me a good introduction. Steve Russell with the Hoosier Mushroom Society. Something I often tell people is that a Hoosier is simply somebody from Indiana. There's no other meaning for the name Hoosier other than somebody from Indiana. Give me one more second. I just want to make sure. Zoom settings. This webinar mode is new to me. Can you guys see the screen?
Yeah.
Trent, can you
no,
just
we
tell
can't
me
see your screen yet. No,
okay.
Not yet. Here, let's let me make sure the screen share is there good for you. Yep.
What about now?
No.
Still no. Okay, give me one more moment.
Mm-hmm.
Let me end the slideshow, find the Zoom settings, share screen. There we go.
Yeah, now we're talking.
Okay.
All right. You
Beautiful.
got it. I'll sign off again.
And so, as I mentioned, a Hoosier is just somebody from Indiana. That's what we call ourselves. There is no other meaning. And for that matter, nobody even really knows where it came from. Why is a Hoosier called a Hoosier? That's still an open-ended debate within the state of Indiana. I'm going to be talking to you today about the next big thing in the mycology, and it's the 2023 edition. So I actually have given this talk, not this particular talk, but a talk with this title. I think it was back in 2017 and 2018. And so the origins of that was the North American Mycoflora project. When that was getting started, I gave a couple talks under this name, but this is going to be the 2023 edition. And several of the things I'm going to talk to you about today are kind of an extension of that general thought process and philosophy that occurred with the original North American Mycoflora project. And so the goals there were essentially to, as the name implies, have a mycoflora for North America. So a survey of all the fungal biodiversity that exists, particularly the macrofungi, the mushrooms that exist in all of North America. And over the last few years, my goals personally have really been to know all of the macrofungi, all of the mushrooms that exist in Indiana. But I've kind of recently broadened back out my personal goals to include all of North America. There's a couple of reasons that I'm able to change those goals a bit. One of them we're going to talk about today, and that's the new sequencing methods that significantly lower the cost of DNA sequencing. And so that's one of the things that's going to allow me to expand that goal back out really to all of North America and not just a focus on Indiana anymore. And so there's a couple slides that I often give in my presentations, and it's like, why am I even interested in this? Why am I interested in the biodiversity at these broad scales? And one of the primary reasons is understanding what occurs, where it occurs, and when it occurs is really foundational knowledge for most further research in mycology. And so whether you're into fungal ecology or applied mycology, like mushroom cultivation, plant pathogens, really understanding what exists in nature and where it exists and when it exists, it kind of forms the basis. Having an understanding of those specific aspects really underpins any further lines of scientific inquiry that you might be interested in going down. And so we still only have a very limited knowledge to this day of the macrofungi that exist in North America. And the more we look, the more we discover. And there's a few things, there's a number of things we'll talk about today along those lines when we start talking about the NAMA specimens. Just a little more about Indiana. Since 2016 is really when we kind of started our local projects. And to date, we've sequenced over 15,000 specimens from the state. We've recorded over 2,500 species. I wouldn't be surprised if that number is probably closer to 3,000 at this point. I haven't done a hard count recently. And so that number is actually somewhat difficult to calculate for a number of reasons. But we've also done sequencing for a lot of different areas and events. Actually over 1,500 NAMA specimens now, 1,000 from the Pacific Northwest. We've done a lot of other different forays across the country over the course of time. But let me start off. I know there's probably a broad range of experience levels on this call. So what I really wanted to start off with is talking about DNA sequencing, but trying to make it as basic and as plain spoken as I possibly can. And I would almost imagine even people that do this regularly might not have thought about it in these particular form. And so the first thing I'm going to talk about today is DNA sequencing. And as I mentioned, this is one of the things that is going to allow us to start to generate a mycoflora of North America, because the costs of DNA sequencing have really come down in recent years. If you think about the first human genome or something like that, it costs something like $1 billion or something, maybe more. It's been a while since I looked at that number for a single genome. And so the cost of a human genome is now down to maybe 500 less, maybe down to $100, depending on the technology that you use. But there's been this exponential decrease in the cost of DNA sequencing over the course of time. And it's about time that that applies to barcoding fungal specimens as well. But when you're talking about DNA sequencing, a single DNA molecule is made up of 4 bases known as nucleotides. So A's, G's, C's, and T's. And a fungal genome consists of about 50 to 100 million of these nucleotides. There's more and less, and that's just kind of a general range. But all DNA sequencing is, all that DNA sequencing means is using some type of technological process, using a technology to determine the order of these nucleotides, of these A's, G's, C's, and T's within the genome of an organism. And so whenever you hear DNA sequencing, that's really all it means, is determining the order of A's, G's, C's, and T's in the genome. And it's really as simple as that. There's a bunch of different technologies you can use to make a determination about the order of those A's, G's, C's, and T's. But at its core, all DNA sequencing means is determining the sequence of A's, G's, C's, and T's within a genome. And so that's slightly different from DNA barcoding. This is another term that you often hear bantered about a little bit, and I'll talk about it in a little more detail today. But DNA sequencing, if you think about the last slide, it said a fungal genome is essentially 50 to 100 million A's, G's, C's, and T's. When people talk about barcoding, that's really only talking about looking at the sequence, the order of the A's, G's, C's, and T's for a very small portion of the entire genome. And these are known as barcode regions. And over the course of time, different barcode regions have been established. They're often 700 nucleotides long. So 50 million in a genome, 700 give or take in a barcode region. But researchers and myself typically use something known as the ITS region, the internal transcribe spacer region, which is right around 700 nucleotides long, to make a determination about which species exist and to identify species and to delimit one species from another. And so I'm not going to go into too much detail of how this process works, but the first thing I want you to understand is the difference between DNA sequencing and DNA barcoding. So DNA sequencing is just determining the order, the sequence of the A's, G's, C's, and T's in the genome. And then DNA barcoding is taking a small barcode region, so a small chunk of the DNA that's particularly useful for making identifications of fungal species, and then looking at that. And so most of the work I do, it's not whole genome sequencing. It's not looking at these very long segments of DNA. I'm really looking at these barcode regions, so these short fragments. And so I would say in 95+% of species that are out there, these these barcode regions are really useful in making species delimitations and identifications. And so what can a DNA barcode tell you? I just mentioned it can tell you, give you an accurate identification. But a DNA barcode by itself doesn't really give you much information. And so if you look on this last slide, that's an example of a DNA barcode. TG, AAAAA, TGAG. This is an actual, I think this is a Lactarius indigo sequence, but you can never identify the organism just by looking at the DNA barcode. So by itself, a DNA barcode doesn't give you much information, and it doesn't tell you much about the identification of a species, or really anything else for that matter. It's just a sequence, a ordered set of A's, G's, C's, and T's that you're working with. But when the magic happens, when you're able to compare that DNA sequence, that DNA barcode, to another DNA sequence and barcode to see if it's the same, or how similar is it to your what's known as reference data. So by itself, a DNA barcode really tells you almost nothing. But in context of reference data, in context of other DNA sequences, it can actually tell you about quite a bit. And so just briefly on how to get a DNA barcode in its very simplest terms, and there will be, there's protocols involved with all of these things, but in its simplest terms, there's 3 steps. You're going to extract the DNA from an organism, and all that means is breaking open the cells to release DNA. So you take a small amount of fungal tissue, put it in a tube, grind it up, use chemicals, use heat. So those are the 3 primary methods to break open cells: heat, chemicals, and mechanical means, so grinding. But the first step is simply to extract the DNA, and all that means is to break open the cells and release the DNA in solution. So once you have the DNA in solution, you're able to take a little bit of DNA and perform a process called PCR that amplifies the DNA. So you take a small amount of DNA, and there's a chemical process. It's a simple chemical reaction that uses a piece of equipment called a thermocycler to exponentially increase the amount of DNA that you're interested in. So if you're interested in the ITS region, which a lot of people talk about, you amplify the ITS region in a simple chemical reaction using just a few basic chemicals in a machine called a thermocycler. And then ultimately you use some form of sequencing technology to look at the order of the A's, G's, C's, and T's in that amplified DNA. And so at its core, that's really all that's going on with any type of DNA sequencing technology that's interested in barcoding, is extracting the DNA, breaking it open, amplifying the DNA, turning a little bit of DNA into a lot, and then sequencing the DNA. So determining the order of the A's, G's, C's, and T's. And that's really, a simplified version of it, but there's, in terms of an overview, that's just kind of a basic overview of the process. If you're interested in this particular subject. So in the past, we've primarily used Sanger sequencing, and that could be thought of as a first generation sequencing technology. I think it was developed maybe in the late 70s, used through the 80s, and we still used it. I personally still used it up until about a year ago for most of my DNA sequencing efforts. And so it's one of many sequencing technologies. Sanger sequencing is using a process to determine the order of the A's, G's, C's, and T's. And that's the primary thing that we used. There are several downsides to it. One of them was that we had to send out all of our samples to a commercial lab for the actual sequencing. The technology itself still is kind of expensive. If you were to try to do it at home or something like that, that's not really a feasible thought process for most people. And the overall cost would have been about $5 to $10 per specimen. That's if you're doing it yourself, right? That's if you're not paying somebody else for labor to do all this. So if you're doing it yourself, it still costs, $5 to $10 per specimen to do that extraction, amplification, and sequencing. And so what we've moved to recently And this is one of the big things, I guess, from the title of this talk, is that we now use nanopore sequencing for all of our DNA sequencing efforts. We've actually moved exclusively over to nanopore sequencing. And so there's a company called Oxford Nanopore Technologies that first released a product in 2014. So it's actually been around for quite a while. You can see the product in the image off to the right. It's a small device, it's portable, it can connect to a laptop, and you can use it at home. And so this is a tool for DNA sequencing. You still have to do all of the other steps I talked about. So the DNA extraction and amplification, that's still required if you're interested in barcoding. But we didn't adopt it up until recently. We only actually adopted it this year because historically it's had a relatively high error rate. So it was first released in 2014. So what are we now? 6, 7, 8, 10 years. We'll be approaching 10 years next year from when this product was first released. And so it's actually been out for quite a while. So I wouldn't necessarily define it as a new product, but it is a new product for barcoding efforts, I would say. People have been using it for a lot of other purposes. But the one key aspect here is that they're able to iterate this product. So every year it keeps getting better and better. So there's different chemistries that are used year to year. There's different flow cells. If you look at the top of that device right there where the pipette tip is actually going in, that's a disposable unit that actually does the sequencing. And so they do iterations of those, they're called flow cells every year. And so every year it keeps getting better and better. And over the course of time since 2014, they've really got it to the point to where it's become a viable option for producing reliable DNA barcodes that we can use for our fungal sequencing efforts. And I'm not going to go into too much detail here either, but the way that it works in a general sense is that you have your amplified DNA. So we talked about DNA amplification. You have this exponential increase in the amount of DNA that you have in your sample. And you do, once again, a basic chemical reaction. You attach something called a motor protein. It's just, you can't see this. This is very, very small, but you just perform a basic chemical reaction and it attaches a motor protein to the end of your sequence. That motor protein guides the DNA sequence through these little tiny pores in that flow cell that we looked at. So on this flow cell right there, right below where they're injecting the sample from the pipette tip, there's about 1,000 of these little pores on that surface. And so the motor proteins guide the DNA to these pores. The DNA goes through the pores and an electrical signal comes out as the result from this process. So if you've ever done Sanger sequencing, you might be familiar with like a trace file. So it's like essentially a raw DNA file that comes from Sanger sequencing. For Nanopore, the raw DNA file is actually a giant file of electrical signals that have to be interpreted by various types of software. And so that's actually another big area for improvement that you're likely to see in the future is that there's a lot of labs that work on different machine learning algorithms to provide enhanced base calling. So you turning those electrical signals into A's, G's, C's, and T's is one of the areas to where Nanopore, I would say, still has a lot of room for improvement. But in a general sense, once again, you have your DNA from your sample, you do a basic chemical reaction, you ligate, that's another word, just attach motor protein to it, put your sample into the nanopore, and your DNA goes through these pores and electrical signals are generated. Some of the primary advantages of nanopore sequencing, the first one, the obvious one I mentioned, is cost. So we're talking about 60 cents or less per specimen versus $10 from Sanger sequencing. And so that's essentially like an order of magnitude decrease in terms of the total cost of sequencing with nanopore process versus sequencing with Sanger. And so anytime you're talking about an order of magnitude cost decrease, that means you're able to run an order of magnitude more samples. And so that's one of the key aspects that's going to allow us to really scale up the number of samples that we're able to process and get DNA barcodes for. If you've ever done Sanger sequencing, if you wanted to do a local project and sequence 1,000 specimens, you're probably talking a $10,000 investment. Whereas with Nanopore, you're talking less than $1,000 to be able to do 1,000 specimens. And so the cost savings is one of the primary drivers to why I'm interested in this process overall, just because it allows me to do so many more samples, allows me to find out so much more important information, and really to start scaling up some of these efforts that were first envisioned with the North American Mycoflora project. Without that significant cost reduction, doing tens of thousands of specimens across the continent really isn't feasible unless you have these massive grants. I think if you think back to some of the original talks, they might even still be online somewhere. For the North American Mycoflora project, some of the first proposals would be like, multiple $1 million plus testing centers around the country or something like that. And at the time, that's absolutely what would have been needed to actually, to make a real stab, a real dent in understanding the biodiversity that exists in North America. But now that nanopore sequencing is beginning to come online, with an order of magnitude decrease in the cost of obtaining these barcodes, you're able to process an order of magnitude more specimens with actually much less time. So there's a number of other aspects. I'm not going to go into any detail here either. We're kind of short on time frame this evening with an hour. But if you're into sequencing, you probably run a lot of gels. You've probably done a lot of sequence editing, manual sequence editing of your Sanger trace files. And both of those things are eliminated with nanopore sequencing. I mentioned it can be done at home. It can be done in short time frames. Another cool aspect is that many types of failed specimens are now successful. And so with Sanger sequencing, let's say you had a bolete with hypomyces on it, and you tried to run a Sanger sequence on that specimen. The results that you get back would just be kind of a muddled sequence. You wouldn't be able to tell the actual order of the A's, G's, C's, and T's, because the raw data is actually two different types of fungi that are superimposed on top of one another, and you can't disaggregate them appropriately. So you're not able to, make heads or tails from the DNA sequence you obtain because there's multiple fungi that are involved. One of the great things about nanopore sequencing is that those specimens are now successful because you'll simply get a sequence file of the contaminant and a sequence file of the target, rather than having them both superimposed into one file. And so that's definitely a huge benefit. And so you often get things that are not successful with Sanger sequences can very often be successful with nanopore sequencing. But there are also some disadvantages to nanopore sequencing. One of the primary ones, something I would really like to get away from at some point in my life, is the amplification process. So where I mentioned you perform that chemical reaction that turns a little bit of DNA into a lot of DNA, that process is one of the things that takes quite a while still, and there's not an insubstantial cost to it, especially when you're doing it at a large scale. But that's still going to be required for nanopore sequencing if your goal is to generate DNA barcodes. Yes, you could do nanopore sequencing without any type of amplification process, but doing so means you're going to be able to do far less samples per flow cell. Remember, the flow cells are those little disposable units that go into that piece of technology that have all of those nanopores on them. And so one of the things that makes this so cost effective is that I'm able to do about 960. I do 10 plates worth of specimens in a run. A DNA plate's typically 96 wells, 96 specimens. And so I currently do about 960 specimens at a time on a single nanopore run. And it's pretty cool because you end up mixing all of the DNA from these 960 specimens together. And then you have to sort that back out somehow. And there's some technical processes involved with that I'm not going to go into today. But overall, it uses lots of specimens in a run. You're able to do 960 specimens at once or more. You could probably double that number and still get good results. But the more samples you put on an individual run, the longer it's going to take to fill that run, meaning you're going to have to wait longer for your results until you have enough specimens to run. But it also presents the possibility for catastrophic failure. So, with Sanger sequencing, if there's one sample that doesn't work, you mess up on one of your cells as you're working through the laboratory protocols, it's not that big of a deal. But when you're mixing 960 specimens together and you're wanting to put them into a process and do all of your sequencing for all of these specimens together, everything has to be basically spot on correct. Otherwise, you have that potential for a really catastrophic failure to where you lose all of your samples. And so, I wouldn't make light of the laboratory protocols and processes that are involved with nanopore sequencing. You do have to have some really reasonable lab skills. People that are thinking about getting into nanopore sequencing, I definitely would not do that until you have mastered Sanger sequencing. So I would still look at Sanger sequencing as kind of a stepping stone for some of these larger DNA, for some of these newer DNA sequencing technologies. So definitely do not start with Nanopore. If you look at my protocol, actually, let me back out of this for one quick second. I do have a full protocol available online for DNA barcoding fungal amplicons with MinION, that's the Oxford Nanopore device, and Fungal. Not going to talk much about the Fungal today, but it allows allows for a much cheaper flow cell. So the flow cells are actually only about $100 a piece with the Fungal. The MinION ones are much more expensive. But there is a protocol online. Let me just drop that in the chat for you guys. If you're interested in checking it out, you can see that it's not a particularly short protocol by any means. I think there's 5, 6 maybe main sections to the protocol. Each of these are multiple pages with many steps to them. 1, 2, 3, 4, 5, 6, 7, 8, 9. So there's actually 9 different protocols that combine this one protocol. So you really need to have some type of lab skills already established if you're thinking about getting into this kind of thing. It's not just something you can go out and buy everything for and expect to have some success the first time, unless you already have some established lab protocols. And so I would just mention, if you're thinking about getting into this or doing your own DNA sequencing, I would kind of say, hit the brakes a little bit on that thought process until you're really readily established with one of the easier methodologies such as Sanger sequencing, to where, with both of these methods, they're essentially the same for extraction. They're essentially the same for the amplification, those first 2 steps that we talked about. The only difference is the DNA sequencing technology. A couple other disadvantages I guess I would mention is you aren't left with a single file at the end of the day, but you're left with a file with, 960 individual sequences. And the pipeline is automated, but it uses 3 or 4 different programs. And so you really have to kind of have an understanding of Linux-based systems, coding in the command line, running commands on the command line. And so that's another kind of secondary hurdle that you need to overcome, another barrier to entry in terms of this whole process. You really need a really good computer, particularly a good graphics processor to do this. And I mentioned the Linux based software as well. And so there are a number of things to consider other than, sometimes you might see online, you can just go and buy this $100 or $1,000 unit that does your DNA sequencing for you. I would guess I probably spent upwards of $10,000 to get this whole thing operational. So it's, even though the unit itself for sequencing doesn't cost relatively that much in terms of, $1,000 for this little unit that we saw a second ago, that unit itself is not very expensive. But then once you start looking at the protocol and everything that's needed, all the chemicals you need, all the, once you start tallying up all the pipette tips, all the tubes, all the pipettes themselves, just everything, all of the laboratory equipment that's involved. I already had most of that laboratory equipment starting out doing this, and I'm still probably up, over $10,000 to get this process started. And so that's definitely something to consider as you're considering this. I didn't even mention the cost of the chemicals. So Oxford Nanopore has proprietary chemicals that you also have to buy. Some of those are $600. They require another chemical from another company that's about $600. And so once you start looking at the whole life cycle of everything you need that's involved in this process, it does become somewhat expensive. But once you have everything, once you're really, set and you're actually getting your sequences in, you make up that cost in a relatively short time, just because of the number of specimens that you can run and the cost that it takes for each individual specimen to actually get your DNA barcodes from. And so that was just a quick primer on nanopore sequencing. And I think, so I actually put this protocol out last summer. And so it's been out in the ether for at least over 6 months now. And I think it's going to be one of the key things in the future, that as the years continue to progress, as Oxford keeps getting better chemistries, making better chemistries available, they keep making better flow cells available, it's only going to get more and more reliable and cheaper and easier and easier as time progresses. And so I guess that's another thing, if you're thinking about getting into this process, this is really kind of in the early adopter phase at this point. And it's only going to get easier as time progresses, and it's only going to get better as time progresses. And so this is all just kind of a version 1 in terms of a thought process and a protocol for nanopore sequencing. But the cost driver is really one of the biggest things for me, as well as the throughput. So I'm sometimes bill it as being able to do 10 times more sequences for the cost for 10 times less work. Once you start taking away the amount of time you might otherwise spend on things like gel electrophoresis that I mentioned on this slide, and sequence editing. It is a lot faster, it is a lot cheaper, but there are also some disadvantages of it that you need to be aware about. Overall, for NAMA, this is Trent showed the image at the beginning of this presentation, to where we were actually ran a flow cell nanopore sequencing run at the NAMA event this last year. So truth be told, it wasn't specimens from the NAMA event that we were actually running in that picture. It was some specimens I brought with me just to be able to show people the process who might have been interested. I actually ran these NAMA specimens from Missouri NAMA 2022 about maybe a week after the event itself. So in that picture, so it would totally be possible to run all the specimens from an event at the event itself. But when you start thinking about the other steps that are necessary, so the DNA extraction and the DNA amplification, those 2 steps require their own set of protocols and time. And so it doesn't really become too feasible to do too much of that at an event itself. It becomes logistically troubling. And so I would much rather do it when I'm in the comfort of my home after the event. And so I would guess that's probably how things would progress in the future. There probably won't do a whole lot of sequencing at the event itself, but instead making the results available shortly after the event, say within a week after the event. So let me give you a quick overview of some of the things we actually saw at NAMA this year that we were able to do some sequencing for with the Nanopore technology. And so we looked at 290 specimens, and we were able to get success rates of about 75%. So we had 219 successful specimens out of that set. That is among the lowest percentage that I've ever had on a nanopore sequencing run. One of the reasons for that, it was really dry, and we took a lot of subpar specimens and attempted to run a barcode on them, and you'll see that here in a second. But if we do a quick overview of all of the NAMA sequencing to date, we're at about, I need to move my zoom button, we're at about 1,500 total specimens that have been sequenced over the years from NAMA to this point. The latest is 2022, Potosi, Missouri, 219 specimens. One other thing I wanted to mention as a part of this is one of the ways that people can help out at these types of events, especially in the future, is actually just going through and identifying the specimens that people bring into iNaturalist. Everybody loves their specimens to be identified, especially if you don't know what it is, or even just getting validation on your ID is often very helpful. And if you look at the top identifiers for the NAMA event, there were only a couple people that did the majority of the identifying. So I would definitely like to thank John and Alan for their efforts during that event, to where we were able to begin, which was really the NAMA Missouri event, was really the first large-scale foray, I believe, that utilized nanopore sequencing within mycology, as far as I'm aware. One other thing I would mention is one of the largest missed opportunities I see within mycology as a whole is actually saving specimens from many of these large organized forays. And so I've been to, I go to lots of forays every year all across the country, and it's always kind of heartbreaking to me every time I see the foray organizers at the end of the event just kind of shoveling all of the biodiversity that was captured at the event into the trash. And that's a common theme with most large forays all across the country still. And I guess that's really one of the largest. So there's not many other times when you're going to have hundreds of people coming into a single event and essentially scouring the woods and vacuuming up everything that's around in terms of fungi, bringing it to one location, to where it's the perfect opportunity for people to start saving some of these specimens. Once you start getting in the habit of saving specimens, that's when you can actually begin to further research them. So taking pictures of the foray is not enough. And we'll see why here in a second as we start going through some of these specimens from the NAMA foray. And so just due to the interest of time, I'm not going to go through a whole lot of them, but some of the key ones that I've found to be interesting, we'll briefly touch on from the NAMA event. The first one is Hydnum subolympicum, and it's probably the most common Hydnum or pandem in Missouri. I saw that Trent mentioned Rachel Sweeney is going to be on here in a couple weeks for a NAMA webinar talking about chanterelles. Well, her previous work was actually this Hydnum paper a few years ago, and they looked at most of the Hydnums from Eastern North America at least, and actually put some new names on them or resurrected some old names for them. And this is probably the most common Hydnum or pandem in Missouri. It's one of the most common in Indiana. It's in the rubescent group, so it will actually stain a little bit red. In Indiana, we have 6 species of Hydnum. I would guess Missouri would have about the same. And I mentioned that just taking pictures of these things might not be sufficient. And this would be a very good example of that. Pluteus hongoi, it's in the Pluteus cervinus group. In Indiana, and I would assume Missouri, only about half of what we might call Pluteus cervinus is actually Pluteus cervinus. The other half is this species. And so if you think about using a species list or images, a species list based on images in the future, if you just have this picture right here and it's called Pluteus cervinus, unless you actually have part of that specimen saved and you're able to perform further analysis on it, whether it's microscopy or DNA, as we were talking about earlier, your species list is never really going to be up to date or accurate, just because there are so many what are known as cryptic species, or species that you're not able to differentiate easily just by looking at them. And the Pluteus cervinus group is definitely one of them, but I would say in almost every genus I've ever looked at, there's many other species that would fall in the same type of category, to where without some type of further examination, the majority of your IDs are not going to be correct. And I would say that's going to be true for any foray. If you were to actually look at a species list that's been generated in the past for the foray, I would estimate that at least half of the identifications from any foray from anywhere in North America, at least half of the identifications are not going to be correct. They're not going to withstand the test of time, just because of what we're learning every time we look at new data sets with DNA and macrofungi. And so that's really one of the top reasons to begin the process of saving specimens, is that over the course of time, your species lists are just going to continue to get less and less accurate. And really, unless a specimen is retained and you're able to get some type of objective measure of what this thing is, it's really going to be required in order to make the most use of the time that you spend taking these photographs. And so if there's one thing I would ask of you as a result of this presentation, and it's kind of the same thing I've been asking for people for many years now, but you'll have an opportunity here at the end of this presentation to participate. But it's really just as you start going out and making collections and taking pictures, the top thing that you can do is to really start saving all the specimens that you find. So even if you don't have the ability, have the funds, have the time to do any DNA sequencing at this point, there's going to be someone like me in the not so distant future that's just going to be asking for general collections from anybody who has any that would like to send in. So that's essentially what we'll be doing this year in terms of the MycoBlitz that we're holding for several weeks this year, which I'll talk about in a moment. But there's going to be a time in the near future to where we're just going to be kind of scouring for anything that's been collected at any point in time from anybody. And so even if you don't have the connections to somebody that can do DNA sequencing, or even if you don't have the funds, if your club is low on funds, that really shouldn't matter at this point, because even if you don't think you'll be able to get the funds, somebody at some point in time in the near future is going to be trying to find as many possible specimens as they can, utilizing the Nanopore technology that we just talked about a moment ago. This is an example of what I meant by many of the specimens that we retained were not in ideal condition. It's the Lactarius indigo. It's lost most of its color. It's lost most of its ID capabilities just from looking at it, but we're still able to get a successful DNA barcode, and we're able to put a species name on this. Just briefly, I would mention, if you look at the name, it's a temporary code name that I utilized in most of my research, Lactarius indigo INO2. There's a whole bunch of Lactarius indigo all around North America. This one has the designation INO2. You wouldn't know that by looking at the condition of the specimen on the right. And for that matter, most people would never save that type of specimen or DNA barcode it. But since we were at the NAMA event, the specimen was available. Now we're able to say Missouri has this particular species. Whenever somebody puts a name on it, we're going to be able to say that the species is also found in Missouri. And prior to this event, we were not able to say that. And so sometimes I'll talk about DNA barcoding quality specimens versus museum quality specimens. This is definitely not a museum quality specimen. Most people would never consider saving something like this, but at a foray, why not? You know, DNA sequencing is cheap enough now. We'll give it a go and see whether it works. In Missouri, they most commonly use the name Amanita bisporidra, the bisporidra group. We didn't find that species at this event, but we found two others, Subaliacea and Amarivarosa, as well as 5 other species of Amanitas. All of these are fairly common to Missouri. Harveyi subcochrae. Harveyi is the Flavoconia-like thing right here. And then several different things, several different lepidellets that are really common in Missouri. A Neobelita species designated Missouri 01. It's also known from Louisiana, but it's the first record in Missouri for it. Several hiddenellums that were interesting. Piperidum. It's also known from North Carolina, Tennessee, Georgia, and Indiana. And that's one of the things that once you start getting DNA barcodes of a lot of things, you're able to start linking these collections with varying morphologies in various locations to start getting a true understanding of the phenology of these species. So their habitat, their range, when they actually fruit. And that's really one of the big advantages by thinking about these things in terms of like a DNA barcoding quality specimen versus a museum quality specimen. I'm totally fine saving a small bit of something that's not in ideal condition because it's going to teach me something. It's going to teach me, this is an obscure look, or this is what this mushroom looks like as it ages, or it's going to really extend the range or the seasonality of this species. And so I take a pretty broad view of what to me is a quality specimen, an acceptable quality specimen to DNA barcode, just because even these poor quality specimens can tell you quite a bit with any individual collection, even if it's not in a museum quality condition. Phelodon mississippiensis, this is the second record on iNaturalist, and the first record was only 22 days before this collection. So that was a pretty interesting one. 19 different species of Russula came into the Missouri foray. 4 were previously known from Missouri, 10 had previously collected in Indiana. We were only able to apply reasonable names to 2 of them, but then I would also annotate I'm not really confident on either of those names, on that being the final long-term application. But when you start utilizing this type of naming process and utilizing DNA barcodes, the specific epithet for a collection becomes less important, because you have this objective measure, this DNA barcode, that's searchable long into the future by other researchers who are looking at these groups. And whether or not you apply the same name or the correct name isn't really that important. The main thing that's important is that you link like collections together, so that everything you're calling Amanita species Indiana 34 represents the same species unit. And that's one of the key things that I work to do with most of my biodiversity survey efforts. It also allows you to keep track of things like how many russulas are there. In Indiana, we're probably around 200 species of russulas right now, which, by the way, probably about 50 of them at least have some form of some shade of red, either on the cap or the stem. Scutellinia is one of the groups of species that are wildly undersurveyed, I would say. So this is Scutellinia species INO1. It's our most common species of Scutellinia from Indiana. But just a short story, this last year, I started seeing a few more species of Scutellinia popping up. You know, we'd have Scutellinia species INO2, INO3, INO4. I'm like, you know, there's something going on here. And so I put the word out to all of our collectors in Indiana, start saving basically every collection that you find of Scutellinia. And now we're up to 17 species of Scutellinia within Indiana alone. And so for a lot of these groups, there's going to be a whole lot of undiscovered biodiversity that's out there. But once you start looking, it's really easy to separate and understand and get an understanding of what actually exists. New Alnicola species from Missouri. Once again, when I use these temporary code names, it doesn't mean that it's new to science. Early on, we talked about the reference data, that the DNA barcode is only useful in the context of reference data. And so I often use these species, these temporary codes, when there's not reference data available for an individual species, but I believe the species to be identifiable based on the barcode. And so this is an example. There very well may be a species epithet that's somewhere buried in the scientific literature, but the type specimen has never been sequenced for this species, and nobody else has done a DNA sequence for a named specimen on this species. And so for my purposes, I'll just go ahead and give it a temporary code until there's reference data available to allow me to link it up to its best name. An alternate type of issue with reference data could be found for Melanoleuca species INO3 from the Nanopore. In this case, there were multiple names that had been applied around the world for this species, and I'm not sure which is correct. So without more investigation, the DNA barcode matched the three names that you see there. Polioleuca communis and Sereniifolia has a worldwide distribution, but there's multiple names that have been applied for this one individual species unit. It could be possible that they're all correct. It could be possible that they're all synonyms. It could be possible that none of them are correct. It could be possible that the best name, the oldest name for this species, the reference data for it hasn't been introduced into the DNA repositories yet. So it could be possible that even though there's three names available, none of them will be the ultimate name that withstands the test of time. And so this is another example of somewhere I might use a temporary code name if there is conflicting reference data that's available when you're looking at the possible matches for your DNA barcodes. 9 species of Cortinarius that were encountered at the NAMA event. We were able to put names on a lot of them. Violacio flavescens matches a name they put on it from Quebec. Cortinarius carrier was first described from Quebec only recently in 2021. We found this at the NAMA event that extended the range for the species. We do find it in Indiana quite a bit, but I believe this is going to be the westernmost collection for the species. Odor triganus, found all over the East. Just a few examples of some interesting collections from the Nanopore in Missouri of Cortinarius. This is the first known record of this particular species. Once again, when I say the first known record based on a DNA barcode. There could be other records that are buried in herbarium somewhere or buried in the scientific literature. There could be names in play for it. So the temporary code doesn't mean it's novel to science. It means it's the first known record with this DNA barcode. Another first known record, a really pretty one, has some vibrant purple colors when you cut it open. Gills are kind of purplish, nice yellow cap to it. Rounded up with Antenema liquescens. Most of you are probably familiar with this species, I would think, but it's only the second record in GenBank, and we just put the first one in there last year. And so the DNA repositories still have a long way to go in terms of having appropriate reference data in them. And the only way to change that is really going to be to go into herbaria and do lots and lots of DNA sequencing of type collections. And the professional community is somewhat slow to embrace that in any, sizable way. So one of my goals with this talk, there were threefold. One was to give you kind of a brief primer of DNA sequencing with Nanopore, then to review some of the Missouri collections. And the final thing I want to talk to you about today is kind of the third leg to this stool of how can we actually start to achieve a North American mycoflora, a continental scale biodiversity survey. Once you start getting into nanopore sequencing, once that starts to become broadly available, the biggest limiting factor for achieving a North American mycoflora as a continental biodiversity survey is not going to be the cost, and it's not going to be the time that it takes to do the DNA and all of that. Historically, those have been the primary hurdles to overcome. It's, it costs so much to do it, and it takes so long to perform the DNA analysis and all of that, even once you have the results. But now with Nanopore, where a lot of the sequence editing processes are automated, where the cost is driven down exponentially, the new limiting factor becomes the number of modern collections that are available. And so if you've ever worked with herbarium specimens, these things can go back hundreds of years. Herbarium specimens, the longer that they sit there, the less usable their DNA becomes over time. And so, the best DNA results are going to be from recent specimens. Things have been collected in the past, 20, 30 years, somewhere in that time frame. Once you start getting older than that, you start seeing really noticeable declines in the percentage of specimens that are successful. And so for these, to be able to get usable DNA from many of these specimens, we really need to have lots and lots of modern collections that are available. And something we've been doing in Indiana, which I would say is far and away our most successful, thing that we do every year, our most, scientifically relevant and scientifically valuable thing that we do every year, are these online forays, where we have people from all across the state. We do a summer and a fall online foray every year in Indiana. We've been doing this since 2017, but we get many people from all across the state submitting many hundreds of specimens. I think our fall foray this last year has probably something like 1,500 specimens that were submitted from Indiana. And this is really the thing that's allowed us to scale up the rate of discovery of biodiversity over the course of time, is really these online forays. And especially with COVID, I'm really surprised that more people didn't pick this up. Everyone was trapped in their homes. Nobody could see each other. And we did lots of them during COVID. We did our normal routine. People can go out in their own homes, on their own property, in their local communities, to their local parks, and take their images out in the field, pick the specimens and dry them. And all they do at the end of the day is mail them to me. And especially during COVID, I was really surprised that more clubs didn't take on this model, just due to the fact that you couldn't go and see each other. But this year, we're really starting to kick this back into gear. Some of you might remember the first attempt at this was in 2019, and that was actually funded by National Geographic. And we got a grant from National Geographic to actually conduct the first Continental MycoBlitz in 2019. And we ended up having about, I think, 2,000 specimens from that event. So it was a very reasonable showing. Lots of you on this call, I'm sure, attended and helped out with that. But we're really going to start to look to do this every year now, I believe, and to really try to scale it up, kind of like we've done in Indiana. If we can get 1,500 specimens from Indiana alone, here in a few years, I could see these types of events easily bringing in 10,000 specimens a year from everybody, from all of the associated clubs, all of the individual collectors. I would think that's going to be a pretty reasonable goal in just a few short years. And so if you think about the needs of what's going to be required to actually have a continental scale biodiversity survey and to actually make it happen, we're going to need collections on the order of 10,000 or more a year that are getting DNA sequenced, DNA barcoded. We're going to need that regularly every year ongoing, right? In Indiana, our initial goal was 10,000 specimens to get DNA sequenced. I mentioned we're about 15 now. I think we're probably going to need, in Indiana alone, probably about 40,000. That's my new revised estimate in terms of goals as to how many specimens we need to have a reasonably comprehensive, not even a comprehensive, a reasonably comprehensive, something like 95% of the species that exist in Indiana. I think we're going to need about 40,000 collections in Indiana alone to get there. And so if you were to scale that number outward to try to guess what we might need in the continent, well, that's going to be a pretty big number. And so if we're ever going to get there, we're really going to need to get started basically now. And as I mentioned, even if, for these events, if I get 10,000 this year, I'm not going to be able to handle all of those myself, if for no other reason than cost. These events aren't being funded by National Geographic this year. They're being funded by the Hoosier Mushroom Society, as well as I think NAMA is going to get involved later in the year, as well as a number of other organizations. Hopefully some of your local clubs might get involved in this too to help distribute some of this cost. But for right now, it's primarily the primary funding mechanism for this West Coast event anyway, is the Hoosier Mushroom Society funding West Coast collections, as well as myself a bit out of pocket. So, I just kind of cover anything else that's needed. But, over the course of time, this is where we're really going to need to get to, is having lots and lots of modern collections that are available. And that's really going to help us to start understanding the phenology of these species, where they occur, when they occur. Remember that first slide that I showed you at the beginning, what occurs, where it occurs, and when it occurs. If we're really going to start to understand that in a meaningful way, we're going to need a significant deviation from historical collecting methods to be able to achieve this. It's just not going to be attainable with the collecting style and the number of collections that have been historically brought in for any type of biodiversity effort. And so, I think a reasonable goal here in a few years, 10,000 plus a year is really what's going to be needed. And then maybe in, 15 to 20 years, we might be able to achieve a reasonably comprehensive mycoflora of North America. And so these are really long-term thought processes, right? It's not like we're going to achieve this tomorrow. It's not like it's going to be done in a few years. This is going to require a decade or two of sustained collecting using a new model for biodiversity survey. And really one of the only ways is to do this online. So, having people get out into the woods using their iNaturalist app to upload it. The way we do it is we have voucher slips to where people are able to have unique numbers for each individual collection, assign that unique number to their iNaturalist record and put it with the specimen, dry it out, and mail it in. So that's really the basic part of the process. What we've been doing in Indiana for 5-6 years now is, people get out, upload them to iNaturalist, dry the specimen, mail them to me with the iNat number. That's it. That's all you have to do for this Continental MycoBlitz. If your club is interested in participating, I'll drop a link here in the chat. I'm going to have to wrap it up here soon, just due to time, but I'll drop a link into the chat with more information for you guys on how to participate. But the question for me, when thinking back to kind of the origins of the North American Mycoflora Project and how do we actually achieve some of those original goals, the question quickly becomes, how can we document the most biodiversity in the shortest time frames at the lowest cost with the highest accuracy? And my answer to that has always been citizen science and DNA barcoding. And, there's not really many other avenues to be able to achieve this, but with the two things I talked to you about today, the nanopore sequencing with the significant reduction in the cost of getting the DNA barcodes, as well as this online foray model, this Continental MycoBlitz model, which will allow an exponential increase in the ability of citizen scientists to make a substantive and sustained contribution. these two things will really allow both of these parts of the equation to scale up exponentially. And I hope that you and your clubs are able to participate in these in the future. And so with that, I think that about wraps up my presentation today. I'm totally willing to stick around at least for the next half hour or so to answer any questions that you may have. And I hope you enjoyed it. If you're interested in, I'm always open to emails. Shoot me an email, steve@hoosiermushrooms.org, if you have any more questions. I believe I dropped a note in the chat. Let me exit out of this really fast. I dropped a note in the chat with the Nanopore protocol. I'm going to drop a note in the chat regarding the Continental MycoBlitz. And if you look at this at the moment, you can see where we are currently with the West Coast Online MycoBlitz as of right now. 1,500 observations, 89 people participating. And so it's going to be a very successful event. It's only something we planned a few weeks ago. We just started
Stephen,
getting out.
can I just real quick, I think you need to share that note,
okay.
that chat with everyone for everyone to see it.
Sorry. Oh, got you. I'll probably need to put it in the Q&A, don't I?
No, the chat works now.
Okay.
I think you just have to select everybody to send it to.
Okay, got you. Give me one quick second. MycoBlitz chat. Moving away from, I'm only able to see hosts and panelists in the chat. I'm not able to select everybody.
Okay, I can do it real quick. I'll send it to everybody for you.
Yeah, if you would, that would be great. So there's probably lots of questions, I would say. So let's start going down the list, I guess. Welcome, Stephen. Hoosiers. Thank you, Brooke. So Gene says it's very unfortunate Steve's distancing himself from fundus.org. I don't mean to. That wasn't my intent in any way. I would say their focus is more on conservation now. If they want to get back into the DNA barcoding game and to really try to do a mycoflora of North America, I'm not distancing myself in any way from them, I would say. How do you determine which barcode area to use? From Peter Kordiak. So when you're making that determination, different groups are going to have different amounts of reference data available for different regions. And so you want to use, if you're looking at a particular group, you want to pick a barcoding region that has the most reference data available for your group. Otherwise, you're going to have to do lots and lots of collections and essentially make your own reference data. And so for fungal groups broadly, you're going to want to use the ITS region just because it has the most reference data that's available. Don Moore, how does the error rate of Nanopore compare to Sanger? So that's a difficult question. It's a loaded question, I guess. From my assessment, I guess you have to understand the differences in the two technologies to begin with. With Sanger, you get one read as a result. You get one DNA sequence. With Nanopore sequencing for an individual specimen, you might get thousands of individual reads, individual DNA sequences. And part of the pipeline that I talked about, the computer pipeline, is you have to merge all of those thousands of sequences together into one sequence. Any one nanopore read, I would say, is not the most accurate, and it's going to have maybe up to 1% error rate. But when you have thousands of individual reads, individual sequences for a collection, and you're able to essentially merge them all together into one consensus sequence, that's where you see a major reduction in the error rate, and you get error rates that are very comparable to Sanger. I would say the majority of the sequences that I look at anymore, they match other specimens in my database that were done with Sanger 100%. So the majority are going to have 100% match from Nanopore to Sanger. Rick Watts, do you have a preference for iNaturalist over Mushroom Observer? If so, why? I primarily use iNaturalist for all of my work these days. I don't actually have a preference. If people want to use Mushroom Observer, if you're sending me specimens with a Mushroom Observer number on it, that's perfectly acceptable to me. I have pipelines built already to accommodate that. For my own work personally, iNaturalist is more user-friendly from my perspective, and they've had the app the mobile app that I've often used for a longer period of time. And so I've moved most of my stuff there, but for most of my work, I don't have for my work, I have a preference of iNaturalist, but if anyone else wants to use Mushroom Observer, I think that's great. You always, Elizabeth asks, Stephen, do you always use ITS1, ITS4, or do you select other primers based on the species you are sequencing? In a general sense, everything gets a first pass with ITS1F, ITS4. I am still kind of making a determination on what I want to use for retries of everything that failed the first time. I'm probably going to switch that up, but I don't know to what yet. ITS1F and ITS4, they're kind of known as the general fungal workhorse primers, and they work great for many things. And so, yeah, 1F and 4 is the primary thing that I use for everything. So there's going to be options in the future probably of doing multiple genes in one PCR reaction, using multiple ITS primers nested in a single PCR reaction. So that's a whole nother topic that's going to take an hour to discuss in and of itself. Rick Watts, what's the value of species concept if so many are cryptic? Well, it obviously makes any one individual species concept less valuable. It makes a morphological species concept less valuable if you can't separate things morphologically. It means you can't use any one species concept and have it be applicable in the DNA age. You have to use essentially a combined species concept. You have to look at all of the information available to be able to make a determination on what you believe to be a species. So yeah, it makes morphology less valuable in some instances. In some instances, it makes morphology more valuable. There's many instances like the Lactarius indigo, as an example that we looked at today. It's easy to separate species Indiana 1 and Indiana 2 just based on their morphology. It's just none of the descriptions are written that way. Every description that you've ever read is probably a conflagration of species Indiana 1 and Indiana 2 together, and no one's ever tried to write a description that separates them based on morphology. But in many cases, morphology is totally possible to see once you see the differences in the DNA. Anonymous attendee, what are the chemicals needed that are proprietary to Nanopore, and how often do you have to purchase them? Fortunately, you don't have to purchase them very often. There is I mentioned the Fungal, there's a Fungal kit that requires all the chemicals for adapter ligation and sequencing. There is another kit from a third-party company that you need to get for, there's a process called DA tailing. That's one of the protocols that's listed. And so I think it ends up being about $50 total per nanopore run in terms of just the chemicals. Rita Burson, what's the best way to save specimens for later? If you go to the Continental MycoBlitz page, it discusses this a bit, but just throwing them in the dehydrator 12, 24 hours, something to that effect will be great. Do you have any resources you can share to look further into fungal sequencing methods? Email me on that. I have lots of resources on all that stuff. It depends on what you're looking to do and what your goals are. Different goals will require different methodologies that are used. So depending on what your goals are, I may have a recommendation for one technology over the other, or one methodology in terms of extraction or amplification over the other. What are the limitations of using the ITS region? There's plenty, and there's whole papers written on that subject, so that's not one I'm going to go into as a part of this. How does age of a specimen affect the probability of success? It totally does. And last question, age in terms of storage of preserved material. Both the condition that it was stored, so if you let something start to decompose slightly or something, if it's not dried well before you begin to store it, and the age, both of them significantly affect the probability of success. The older they are, the less likely they are to succeed. You start seeing significant decreases in success rates for the ITS region, maybe around 20 to 30 years. After that, you start seeing, kind of a constant slope downwards in terms of the success rates. Even up to 30 years, you'll still see differences between recently collected stuff and older stuff. Grace asked, in your experience with nanopore sequencing, has there been a time when the results of DNA sequencing compared to morphological analysis of some fungi that have yielded drastically different results and not just due to error? Yeah, I see that every day, right? So that's one of the things that keeps me going in this overall process is you make new discoveries every time you look at a data set. You look at species that you thought you knew in an entirely new way. You look at novel things that come in. You find new genera with some regularity that you've never heard of. I've been doing this for a while now, and I'm still every now and then I hear of a new genus of macrofungus that I'd never heard of before. It keeps you up to date with the nomenclature as well. You start seeing new names of new genera that might appear in GenBank. So with my previous comment, I was saying, this every now and then I come up with genera that might have been around for 100 years I'd never heard of before. But then the inverse is true. It helps to keep you up with current taxonomy, because then when you're looking at results and you see a new genus name, it gives you the opportunity to investigate it further. Deborah, if saving specimens for sequencing, is it vital to have voucher level documentation of species, location, habitat, etc.? Yeah, obviously it's always preferable if, as you mentioned, my minimum metadata is collection date and location. So if you fill out an iNaturalist observation, you're going to have the metadata that's most important to me. All the rest is just gravy, in my opinion. How can you track the number of sequences described of a species in a region? So the easiest way is to get on GenBank and BLAST, do a search of whatever species you're interested in, and then see if the authors of any matches annotated the location. That's really the only way to do it. What is a species? How do they differ? Do they need to be, how different do they need to be a separate species? Well, that's kind of the question. There was a talk Bryn Denninger gave recently where he said a species is whatever an author wants it to be, and that's essentially my opinion. I'll leave it there. And that's kind of the classic problem within all of taxonomy is how do you define a species? Sarah asks, especially for taxa that haven't been studied. Give me one second. I'm just okay. Yeah, especially for taxa that haven't been studied well previously. Have you ever found ITS to be not helpful, either underestimating or overestimating species diversity? For genera, not so much. And so keep in mind, I don't look at many ASCOs as an example. I look at a relatively small number of ASCOs. But for something like, we talked about eyelash cups, Scutellaria. The ITS seems to be perfectly good at delineating most of the species in those groups. I find it to be less helpful in isolated circumstances within groups. So as an example, say Agaricus, there's a couple species in there that have very similar ITS sequences, and authors believe them to be separate. And I'm not going to argue with that. I think that's probably going to be, you'll find that to be the case because of convergent evolution or other factors that are involved. But yeah, you will find that from time to time, but I would estimate it to be less than 5% of species that are out there to where ITS is not as valuable as other genes may be. You also have to keep in mind, ITS is often not helpful for producing phylogeny. So understanding the long-term relationships of these species, right? Understanding the family tree. ITS is less helpful for that. But if you don't care about that, if you don't care about the phylogeny, if you care about how things are today, it's easy 95% in terms of delimiting individual species. You don't have to have a resolved phylogeny to delimit species. Anonymous attendee, how's an epitype established to use as reference data? There's a process for that in the botanical nomenclature. It's not very difficult at all. If we could talk about that, that's why it's been kind of one of my side projects I'd like to get to at some point is, essentially documenting the type, location of everything, and then sending out all the clubs to find individual epitype candidates. So yeah, if you're interested in that, that's a great place to start. To first start looking at the botanical nomenclature. It lists what is needed to establish an epitype, or you can just look in the scientific literature for what anyone else has done to establish an epitype and model your work after that. Steve Ness accuracy, are there base pairs that just get read wrong by the machine or computer, or how do you know? Well, so at this point, you don't really know. The only way, and the same thing's true for Sanger sequencing. You don't know if any individual base that you're looking at is an error or not, just by looking at the sequence. The only way that you can even guess that it's an error is by having reference data that differs. And so I would say even for most Sanger sequences, nobody uploads their raw data for Sanger sequences, so you can't really trust outright on its face any Sanger sequence that's ever been produced in GenBank in terms of making these comparisons, unless you're able to examine the raw data, which nobody puts their raw data from Sanger sequences into GenBank. And so you can't really say, yeah, most people talk about Sanger as a gold standard, and I would agree that it is in a general sense, but you can't start making you can't say, oh, this sequence in GenBank is, and this is true for phylogeny or species delimitations, or you can't just accept any sequence in GenBank as right on its face, because if you've ever edited a Sanger sequence, you realize how much subjectivity there is in making those base calls for Sanger sequencing. And so that, I would say that's one of my biggest problems with GenBank in a general sense. I have lots of them, but one of the biggest ones is that they don't require the raw data submission to go along with sequence submissions. And without that, the only way you can actually validate any differences is by going back to the original specimens, if they exist in some herbarium somewhere, and sequencing yourself and looking at the raw data. And so, yes, it's possible for a sequence. And so you have to remember what we talked about with Nanopore. It's not looking at bases. Nanopore is making an interpretation of electrical signals as a DNA sequence is flowing through a pore to then use a machine learning algorithm to interpret those electrical signals to call the base. And so that's something that even 100 years from now, the Nanopore data that we're obtaining can still be reanalyzed, and you might get different results 100 years from now because the machine learning algorithms have improved to a point to where they can much better analyze the electrical signals that are being produced. And so getting the data now isn't just a one-off. I fully expect that I'll be reanalyzing certain parts of my data 20 years from now with more modern algorithms at that time. So Nanopore sequencing is a bit different than Sanger from that perspective. You're not stuck with this data in perpetuity. You're going to be able to reanalyze this data in different contexts over the course of time. How different would a barcode, Irene, how different would a barcode have to be to consider it a new species? So there is no set number. Everyone always wants a magical number for that. I have methods, that's a whole different talk. I would say I have kind of novel interpretations of how to do this. But there is no, it's easy to say, there is no set number. If you look at Agaricus that we were talking about, different species can be 100% identical for the ITS region, or different species, the next closest species could be 10% off. So there is no single magic number. It differs for each individual species. Will Beeson, have you found any interesting contaminants in your samples? Yes, all the time. I database every contaminant that I believe to be a biological contaminant on a sample. I have entire databases now of things that decompose amanitas or puffballs, or I save all of that data and index it in my local blast database. And so, yeah, hygroscopies are another one you get lots of interesting contaminants on. So yeah, I have entire databases full of data of contaminants for all of these individual groups. Max asks, is there a DNA barcode set that is universal amongst fungi, or is each barcode set distinct for all fungi? I mentioned that ITS1F and ITS4 primer combination is what I normally use. That works well among most fungi. You're going to be hard pressed to find a much better one. But there are individual groups that ITS1F and 4 combination does not work particularly well for, and there are certain species within groups that ITS1F and 4 does not work particularly well for. And so I would say start there with that primer combination and then move more broadly. Ken, when you do 4-gene sequencing, do the sequences get assembled into one at the end, or are they considered separately? Well, so I guess that depends on what genes you're sequencing. If you were to sequence small subunit, ITS large subunit, you would probably want them all assembled continuously because they're continuous in the ribosome. If you were sequencing EF1 and RPB2, you would not want them conjoined. And so you could do it either way. Liz asked, what's the best way to preserve specimens? Dehydrate them. Steve Ness, have you done any whole fungi genome sequencing with MinION? I have not. How do you exclude DNA pollution from other species? Example, other specimen spores. You don't have to. Cross-contamination is not really a concern. Has barcoding changed the definition of species? I'm sure it has, and I would say that's what a species is a still fluid discussion and an ongoing discussion within all of biology. So there is no single definition of species. Sarah asked, if I throw some money at you, want to sequence 300 specimens from the Iowa Fungal Biodiversity Project. We've worked together in the past, so let's talk. All motors use energy. How does motor protein get its energy? I don't know. I have the sense that it's more of a process. So you're essentially ligating this motor protein. So I don't know whether it's best to call it a motor protein or a guide protein. I believe the literature calls it a motor protein. I think of it as more of a guide protein that doesn't have, essentially something to attach to the pore to guide the DNA through. I don't look at it as like an active process that requires energy, but I just don't know enough about the specifics to comment further there. Will you elaborate on some of the process of comparing multiple specimens, DNA sequences to reference data using Oxford? Elaborate on some of the process of, that's going to be a whole nother talk. So that's essentially a talk on analyzing DNA sequences. How do you interpret them once you have the results? That's a very long and very subjective process, I would say. Next question, what will be the next technology improvement? I think, as always with Nanopore, it's going to be better flow cells and better chemistry. So those are the combinations that they're able to work with. I think over the course of time, that's going to keep getting better and better. Can you lay out the main important points of how to safe dry specimens sent to you? Put them in a dehydrator, 12 to 24 hours until they're crack or dry, put them in a Ziploc bag, write the iNaturalist number on the exterior of the bag. Any reference for specifics on the best way to preserve specimens? Same question. Is there a breakdown of number of reads for different levels of quality? How many is too few versus how many is not great? How many is great? That's actually an interesting question, as well as a difficult one. So I've had 100% matches to reference data with as few as 5 reads with Nanopore. Anytime you're only getting like 1 or 2 reads from something, it's usually not the target. It's usually something else spurious. So those are hardly ever right. Those are hardly ever on target. I've had it go down to as few as 5 reads to have 100% matches to reference data that were on target. That were where you were. It was a sequence of the target specimen that you see pictured. The process that I utilize, my pipeline, it maxes out at 500 reads. So the closer you get to 500 reads as a part of the consensus, is the better it's going to be. Will there be a BioBlitz on the East Coast? Yes. Summer Continental MycoBlitz, August 11th to 20th. Fall Continental MycoBlitz, October 13th to 22nd. Isn't there a maximum temperature the hydrator can be at for the preservation of DNA? In short, no. The longer you keep it at a higher temperature, the more DNA is going to be degraded. But for recent specimens, that doesn't seem to be that big of a deal. I've had stuff that's been maxed out, burnt, almost burnt, right? So using just some old dehydrator that has no temperature setting, left in for 3 days, it works fine. So, I mean, in a general sense, you want to maintain the lowest temperature on the dehydrator. Also, when making a collection, do you have advice on how to prep a sample beyond that? I briefly mentioned like a barcoding quality specimen versus a museum quality specimen. And I think in the MycoBlitz protocols, it talks about it. If you have a large specimen, I only need like a quarter cap. I definitely don't need like the whole specimen. I'm not trying to make museum quality specimens here. I'm trying to get images associated with DNA barcodes, but also to have some bit of the specimen left over in case it's interesting enough for microscopy. So in a general sense, a piece the size of a quarter is going to be fine for a large specimen. If that's all you want to save, that's better than nothing. How will your sequence results be available to the general community of mycologists? They're all available right now. If you went online at mycomap.com and you went to DNA research, mycoblast search. I will drop this link into the chat. You can search my database at any time you want. New search. Give me 1 second. You can do a blast search there for my database, and it is publicly available. All of my data is always publicly available all the time. So there's nothing that I keep private in any way. All of my data is public on the web. Is an ordinary kitchen dehydrator okay? Yes. Thank you, Mariah, for putting that in the main chat. Are there any current progressive government, business, or college forestry researchers interested in helping fungal identification of ecology and fungi to forest health? No, I mean, not that I'm aware of in any real degree, really. So the funding for mycology is very, very limited for the type of work that I'm interested in. It's even more limited. There's probably some stuff out there for fungal ecologists to do Illumina sequencing, which is more or less the same as Nanopore, different technology that's kind of a second generation sequencing technology that has better accuracy than Nanopore. I would not use Nanopore sequencing for environmental DNA at this point. It's just not accurate enough. And so it will be in the coming years, I would say. But as of right now, I would only use Nanopore sequencing for fungal amplicons that are specimen based, to where you're able to look at the actual physical specimen to make a determination. A lot of times when you're doing environmental DNA, you just don't have enough reads to be able to get a great consensus sequence. So I would not use Nanopore for environmental DNA to this point, but I would use Illumina, and there might be some funding available for that kind of thing. But overall, there's very limited available. Amy asks, how did you come up with just using 700 base pairs as enough to differentiate? I didn't. This was first developed back in, I want to say, early 90s. I think Tom Bruns and a guy named White were the first to come up with the ITS-1F and ITS-4, well, at least ITS-1F primers, maybe. I'd have to look back at that paper to see who came up with that method, but it's something that's been commonly used for decades now within fungi. I didn't come up with it. It's kind of a standard barcode region for fungi. Every organismal group has kind of a standard barcode region to where professionals have looked at different regions of DNA and made the assessment that some are better than others in terms of species identification and delimitation. And Trent, I think I beat your $5 buzzer, or 5-minute buzzer. I think I answered all the questions. So if anyone, can mycelium be tested? Asked Trent, and the answer is yes. I do that with some regularity. If I see, a bit of mycelium on the ground or something like that, sometimes I'll just save a bit just to see what species it belongs to. And we found a number of interesting things that way. There's some truffle type species that will actually produce a yellow mycelium commonly. And so now there's certain types of mycelium that you can identify trail side just by the look of the mycelium without ever having to see a fruit body. Another question from the chat, are people using the same thing with plants? I don't know of anyone that's collecting dried specimens from iNaturalist users for DNA sequencing, but yes, it's the same type of process in terms of DNA barcoding with any organismal group. You just have to find the right primer combination where the most reference data is available for your group.
All right. Thank you, Stephen. That was a fabulous presentation. There's a lot of people saying thank you in the chat. I hope you see that.
Yep.
I think we just really appreciate all that you're doing in this world around your protocols and your testing and sharing it with all of us who are enthusiastic about it. But maybe like me, a lot of it goes over my head, and you really make this more accessible, and I appreciate that. And I'm also grateful that there's opportunities for even like mushrooms that I collect to be sent in maybe through the MycoBlitz or the BioBlitz and get it tested and get some hard answers on what it is. So I just want to say I'm really grateful for all that you're doing in our myco community here.
Yeah, no problem. And, you know, I know it's a difficult oftentimes when you're talking about DNA sequencing technologies and methods and extractions and amplifications and all that, it can become quite intimidating for people. I try to make it as simple as I can, but it is kind of inherently a technical topic, right? It's a highly scientific and technical topic. And so I try to do the best I can. But really, you know, especially with things like the Continental MycoBlitz, most people don't need to worry themselves with any of those topics, right? All they need to do is bust out their dehydrator, take a couple pictures, upload it upload it to iNaturalist and write your number on a Ziploc bag and throw it in the mail. And so, you know, people can make a contribution doing just something that simple. They don't have to worry themselves with the DNA analysis or, you know, the technical aspects of how to generate a DNA barcode.
Okay. Well, and also I want to thank you for really helping out NAMA so much in 2022 at our events, and now speaking to us through the webinar, and hopefully being involved in the future as well. We're just again grateful for all you do in this space. This would be a great time for you to share kind of your contact information one more time. I think maybe email and website type shout out here.
Steve at HoosierMushrooms.org. I dropped it in the chat, so actually I can't drop it in the chat. You're gonna have to copy and paste it over or something, because I'm still not seeing getting the option for. That's strange, but Steve at HoosierMushrooms.org is my email address. Feel free to contact me there on any topic for any reason. I'm definitely open to having conversations with anybody, and especially if you're interested in getting involved in DNA sequencing. I love to have those conversations with people. There's just not that many of us that do it. And so I would love to get more of you interested in those kind of things and help out in any way I can.
All right. Well, thank you. You can go ahead and turn your screen sharing off here, and I will end the presentation and say goodnight to all of our attendees. Thank you for joining us this evening.
Thanks.
Bye-bye.